Hello viewers. Today we will talk about the plant hormone abscisic acid or ABA. ABA controls many plant processes including stress responses, development and reproduction. ABA is synthesized partly within plasmid and partly within cytosol. The precursor molecule is the plant pigment zeaxanthin. In plastid, zeaxanthin is converted to xanthoxin, and then xanthoxin is converted to bioactive ABA within the cytoplasm. The bioactive ABA can also be reversibly conjugated with glucose to generate a glucosylated storage form, or it can be catabolized to inactive forms like 8'-hydroxy ABA or phaseic acid. In the biosynthesis part, several enzymes play critically important role, such as ZEP and NSAID within the plastid and ABA2 and AAO3 within the cytosol. Several stereoisomers are possible for ABA, where the S-cis ABA is the naturally occurring active form. Now, the precursor zeaxanthin is present within any green tissue, but it can be limiting during synthesis in roots. Root is the primary site of biosynthesis of ABA. Now, ZEP loss of function mutants are deficient in ABA. ABA is responsible for stomatal closure during water stress to prevent water loss. So, ZEP mutants have low ABA levels. Consequently, these plants cannot close stomata upon water stress such as drought conditions. On the other hand, if we overexpress NSAID, then we have a very high level of ABA in the plant. These plants have enhanced drought tolerance as well as increased seed dormancy, that is, very low frequency of seed germination. This is another important function of ABA. Similarly, if we have loss of function mutants of ABA2 gene or AAO3 gene, then the endogenous level of ABA goes down. ABA2 loss of function mutants show improper germination, such as germination under inappropriate conditions such as salt stress. On the other hand, AAO3 mutants, they are wilty. Conditions such as availability of water or developmental signals, they control the balance between the biosynthesis of bioactive ABA versus its reversible conversion to the glucosylated storage form or irreversible catabolism to 8'-hydroxy ABA and phaseic acid. This ultimately dictates the overall level of bioactive ABA within the plant. If a leaf is detached, dehydration condition starts. In this situation, ABA rapidly accumulates in the detached leaf because this will induce stomatal closure, prevent loss of water, thereby prevent the drying of the leaf. Now, if this detached leaf is again immersed in water, there is a rapid decline in the bioactive ABA because it is catabolized to phaseic acid. The enzymes responsible for catabolism of ABA, that is the 8'-hydroxylase that converts it to 8'-hydroxy ABA, and a cyclase that converts 8'-hydroxy ABA to phaseic acid, they are induced upon rehydration. Similarly, there is an effect of seed germination if there is abnormally high ABA level by loss of function mutation in the 8'-hydroxylase gene. These mutants show very low level of seed germination, that is, their dormancy period is highly increased due to the enhanced ABA level. ABA can be glucosylated by the enzyme ABA-Glucosyl transferase, and this glucosylated form can again be reconverted to free ABA by the enzyme beta-glucosidase. Now, glucosylated ABA is a storage form within the vacuole, as well as it is a transport form through the xylem along with free ABA. So, conditions like osmotic stress induces the glucosylated ABA to be converted to free ABA, which is then transported and can exert its effect. In a well-watered plant, the ABA level is low. Consequently, the stomata is open and there is normal transpiration. But if there is water stress such as drought condition, that induces biosynthesis of ABA in the root, then the ABA is transported to the shoot and ultimately to the leaf, where it causes closure of the stomatal pore and reduces the rate of transpiration and thereby the loss of water. The movement of ABA under water stressed condition has been mapped by a reporter gene based assay, where an ABA inducible promoter is fused with the luciferase gene, and it was tracked that the ABA level after water stress becomes gradually high in the veins and then in the GERD cells. ABA is a weak acid. So, when it is in the cell wall where the pH is slightly acidic, it exists in a protonated form ABAH. Now, this uncharged form can enter the cytoplasm where the pH is 7. In this pH, the ABAH dissociates its proton and becomes ABA minus. Now, this charged form cannot go out of the cell. This is a mechanism to retain ABA within the cell. The transport of ABA is highly enhanced by different type of transporters. For example, there is a particular ABA exporter which is transcriptionally induced in presence of ABA. In this reporter gene system, the promoter of the transporter is fused with Gus reporter gene, and we can clearly see that in presence of ABA, the exporter is highly expressed. Another ABA transporter is present in the GERD cells, which is very essential for the ABA to enter the GERD cell region. Now, loss of function of this particular transporter gene causes reduction in the ABA-induced stomatal closure because ABA cannot exert its effect. Consequently, these mutants are highly susceptible to water stress conditions such as drought compared to the wild type control. The signaling mechanism induced by ABA has been worked out to a large extent. ABA has a cell surface receptor which upon binding to ABA gets activated and then sequesters a particular protein phosphatase. Now, these protein phosphatase when free acts as an inhibitor of downstream kinases induced by the ABA signaling pathway. So, when this protein phosphatase is sequestered, the kinases gets activated. These then trigger calcium independent as well as calcium dependent signaling pathways that ultimately activates different types of transcription factors, which in turn then induce the transcription of many target genes, which result in the observed different ABA responses. These transcriptional targets include different types of genes, including signaling genes, stress and dehydration induced genes, seed specific genes, as well as genes involved in ABA metabolism. Now, in presence of ABA, there is dramatic effect on stomata. When there is no ABA, the guard cells are turgid because they accumulate a lot of water. Consequently, the stomatal pore is open and there is normal rate of transpiration and exchange of gases. In presence of ABA, the guard cells lose their water to a large extent, become flaccid, resulting in the closure of the stomatal pore and reduction in the rate of transpiration as well as gas exchange. These pictures show that in absence of ABA, how the stomatal pore is open, and in its presence, how they start closing. Now, when ABA is there, it causes the ion channels which export potassium ions, chloride ions and many other ions to get opened. At the same time, ABA inhibits the proton pumps present in the guard cells. All these lead to depolarization of the guard cell membrane. Now, since these ions are going out of the cell, there is an increase in the osmotic pressure just outside the guard cells. Consequently, to balance the osmotic pressure, huge amount of water flows out of the guard cell to the exterior. As a result, the volume of the guard cell shrinks and the stomatal pore closes. When ABA is not there, the proton pump is reactivated as well as the ion import channels are activated. Consequently, there is membrane hyperpolarization and huge amount of ions enter within the guard cell cytoplasm from its exterior. As a result, the osmotic pressure within the guard cell cytoplasm increases, and to balance it, huge amount of water flows inside the guard cell from the exterior. Consequently, the volume of the guard cell increases and the stomatal pore opens. Now, when there is ABA, the cell responds by increasing the amount of calcium ions in the guard cell cytoplasm. This increase is exactly parallel with the decrease in the aperture of stomata. Now, in presence of ABA, a special response is observed within the guard cells. That is a rapid increase in the concentration of cytosolic calcium ions. Now, this pattern of increase is exactly parallel to the closure of the stomatal aperture, indicating that calcium ions may play a role in controlling the movement of the ions and controlling the activity of the proton pump, resulting in the membrane polarization or depolarization, which ultimately controls the osmotic pressure as well as the stomatal water content, ultimately leading to either stomatal pore opening or stomatal pore closure. In reality, it has been found that calcium ions act as a second messenger towards ABA. Now, in this diagram, the role of calcium ions has been depicted. So, when ABA binds to the receptor on the guard cell surface, the receptors get activated and then it triggers two different pathways. One is the ROS pathway, another is the IP3 CADPR pathway. Both pathways increase the calcium ion concentration within the cytoplasm, either by importing calcium from outside of the guard cell or by releasing the calcium stored within the vacuoles. Now, this increased calcium ion concentration then ultimately regulates the ion channels. First of all, it closes the potassium ion input channel and opens the potassium ion export channel. At the same time, the calcium can also open up the chloride ion export channels. And during this time, it also inhibits the proton pump present on the guard cell membrane. This results in the membrane depolarization observed, as well as due to the export of the ions, there is increase in the osmotic pressure outside the guard cell, which will ultimately trigger the water release and the corresponding change in the stomatal aperture. Now, water stress, which increases the levels of ABA, has a very interesting effect on growth of the plant. Increased ABA levels inhibits the growth of the shoot. At the same time, it increases the growth of the root. So, we have a relatively shorter shoot and relatively longer root. This is very logical because this will increase water uptake by roots, as well as this will decrease water loss from the shoot, which is essential during water stress such as drought conditions. It has been observed that ABA can induce genes that are very important for protection of cells from desiccation damage. These genes include aquaporins and ion channels. Aquaporins will lead to import of water within the cell, and ion channels will increase import of ions within the cell. That is very necessary during water stress or desiccation condition. Other genes of this class are chaperone genes, which will protect the proteins to maintain their active conformation during this condition of stress. Similarly, osmoprotectants are also induced. These includes sugars and many other amino acids. Moreover, some genes that are very important to defend the cells against oxidative stress are also induced. These include peroxidase and superoxide dismutase. Abscisic acid also play a very important role in the development of fruit as well as its ripening. It has been observed that before there is a peak of ethylene synthesis, there is a peak in ABA synthesis. So, this peak precedes the ethylene peak. In reality, both ABA and ethylene has a role in the development and ripening either at the onset of ripening or during ripening of different types of fruits. Now, fruits can be classified as non-climacteric or climacteric. Now, for non-climacteric fruits, ABA plays deciding role in its ripening, such as grapes and citrus fruits. For the climacteric root, the role of ripening goes to ethylene. The relative levels of ABA and gibberellin can control the fate of seed. When ABA level is high, we see embryonic patterning, accumulation of reserves, as well as desiccation tolerance towards the seed. This is for seed maturation, for seed dormancy, and obviously desiccation tolerance. But when ABA level goes down, then we have reserve mobilization and cell expansion. At the same time, we have an increase in the ABA catabolism and increase in the gibberellin biosynthesis. This leads to the germination of the seed. There are recent observations that indicate that ABA-induced stomatal closure can block entry of bacterial pathogens through stomata. Thus, ABA can defend plants against bacterial infection, making the plant resistant towards some bacterial pathogens. So, it may be possible that when the plant senses that there is pathogenic bacteria in the leaf close to the stomata, then ABA is induced and there is stomatal closure blocking the entry of bacteria and thereby blocking the infection by the pathogen. The signaling pathway of ABA, or so-called the ABA response that results from it, interacts with many other hormones. Some hormones, they promote the ABA response, whereas some hormones, they act antagonistically towards ABA response. Hormones that help or enhance ABA responses is auxin. Many other hormones, they antagonize this. These include brasinosteroids, gibberellins, and cytokinins, which have opposing effect of ABA. Now, depending on the several physiological effects of ABA, many methods of bioassay of ABA has been developed. The name abscisic acid comes from the fact that it was first observed to induce abscission or leaf dropping in plants. This makes perfect sense because leaf abscission will lead to rapid decrease in the water loss. So, this is used as a method to bioassay for ABA. For example, excised abscission zones or explants can be put into solution containing ABA, and the time taken for abscission can be measured and compared with a control where there is no ABA. The increase in the rate of abscission or reduction in the time of abscission is an indicator of the presence of ABA, and this has a quantitative relation with the amount of ABA. Another effect of ABA is inhibition of shoot growth. Now, when we discussed auxin, we discussed a bioassay method for measuring increase in the coleoptile section growth in presence of auxin. Now, in this condition, if we add ABA or a sample that contains ABA, then the rate of increase in the growth of the coleoptile section is decreased. So, this decrease in the rate of increase is a measurement of the amount of ABA present in the sample. Now, since ABA decreases anti-transparent activity by reducing stomatal closure, this can be used as a bioassay method. So, in presence of ABA, the stomatal closure is measured microscopically, and this can measure even very, very low level of ABA. Now, there are several practical uses of ABA. Since it is an anti-transparent, its application can reduce the rate of transpiration in plant by up to 56% in field. Consequently, this can conserve water as well as this can reduce the requirement of irrigation. Similarly, in some short day plants which are kept under inappropriate photoperiods, ABA can induce flowering. It is also extensively used as promoter of rooting in different stem cuttings. Another major use of ABA is prolonging the period of dormancy in buds, in storage organs, as well as in seeds. Now, there is intensive research to develop drought-tolerant plants all over the world. Because of the developing water crisis throughout the globe, this is a very important research. We need to know in much more detail how ABA contribute to the development of root, uptake of water, rate of transpiration, as well as the desiccation responses of the cell. Now, if we can understand the detailed mechanism of ABA responses, then probably we will be able to answer many important upcoming questions, such as how do guard cells integrate so many different signaling pathways, or what actually controls the activities of the ion channels in the guard cells. Similarly, how seeds can integrate ABA and multiple other signals, or why does expression of stress response genes lead to reduction in growth as well as in the yield? Maybe all of this could answer the most important question, that is, can we breed plants that are drought tolerant as well as pathogen resistant? Thank you very much for listening.